This session, I'm delighted to host this particular session today because it really focuses on some of our successes and a real future-looking view of some of the successful team science projects on campus, what they have been up to, how they've been able to bring teams together around certain topics, and what the future holds for this. So we have a very, very exciting lineup that starts with my colleague, Dr. Baram.
Well, thank you for being here, and it really is exciting to be here and talk about team science in UCI Brain. Before I go any further, can you hear me? You probably can't see me because I'm small and the podium is high, but can you hear me okay? All right, so when Mike asked me to talk about the Conte Center and team science in Conte Center, the question is, what does the Conte Center bring to the table? Well, the study of the brain in this amazing three-dimensional computational mechanism or structure is daunting enough. But what the Conte Center brings is actually bringing the fourth dimension, the notion of development. And what I'm showing you here is simply what happened in prenatal life in the human brain. And the running tally is simply the number of cells that are developing in the brain. And of course, the changes are amazing. So the Conte Center really focuses on brain development, normal and aberrant. And the first phase of the Conte Center went from prenatal life all the way to adolescence. And the second phase, what we just started, as you've heard earlier, really takes principles of brain development into adults, and particularly not only adult mental and cognitive health, but also adult psychopathology and mental illness. And so to understand such complex issues as the contribution of experiences that happen early in life to brain development and how that may impact not only brain health, but also brain illness, really requires what, as Mike said, a cross-disciplinary team and cross-disciplinary teams that are composed of individuals trained in different areas of expertise. Why? Because they bring to the table diverse perspectives, conceptuals, and skills. This allows that kind of team to tackle very complex and major problems, health and world problems of other kinds that arise from many, many different sources. It allows such a team to each bring a new idea to the table and to rehash them and to think and conceptualize them in a manner that provides really novel and comprehensive solutions that are not possible within each discipline. And the outcome of that is you can come up with a solution not only to the problem at hand, but also solutions that may be generalizable and applicable broadly to other world problems. So the return on investment here can be huge. So in the Conte Center, we have a variety of disciplines. We have neurobiology. Some have the mouse. Oh, here's a mouse. We have neurobiology, and I'm showing you some pictures here. I'm there as well. We have a neurodevelopmental psychobiologist and psychologist who have been following fetuses all the way to adulthood. We have a large contingent of computational and quantitative people, led really by Professor Hal Stern, Vice Provost of Academic Planning, that spoke earlier today, and Ali Motazavi and Dave Keeter, who brings different expertise to the table. We have neuroimaging represented, and you see friend Mike Yassa here. And we have a number of clinicians, including, of course, Ruth Benka, the chair of psychiatry, and others. And of course, like any team science, and I can't overemphasize that, we need a key, a linchpin, a director of operations, and we're blessed to have Dina Jankowski do that. So what are we really studying? Well, our approach is to take and introduce principles of developmental neurobiology into both the concept and the study of the origins of mental illness. So we're really starting with real-world clinical issues. We are taking them to the lab, either human lab or animal model lab, and then our goal is to take our discoveries back. And we're focusing on a very novel idea, and the idea is that in addition to genetic, chaotic environmental signals can disrupt the normal maturation of brain circuits because they do not provide them with the appropriate rhythms that allow brain circuits that underlie things such as emotions and cognitive function to develop normally. That leads to circuit dysfunction and vulnerability to disease. In fact, we're looking across species and using the capitalizing on the strength of animal experimental models versus human to look if unpredictability of signals from the environment and from the parent influences circuit maturation and mental and cognitive health. This requires a lot of tools and a lot of new tools. Some of them are mathematical and quantitative, and the quantitative and computational portion of our team science has really been able to quantify human behavior in terms of entropy rate, which is a quantitative measure of the degree to which a future behavior can be predicted from current behavior, led by Hal Stern. And that allows us to really look at chaotic and unpredictable signals and really establish that they may not allow normal maturation of brain circuits. In the current iteration of the Conte Center, as you heard, which has been recently funded, we go not only to assessing behavior in human infants and understand mechanisms in animal model throughout development, but we are taking this all the way in a trajectory to really understand vulnerable adult populations and how chaotic early life events may influence their mental health. So again, to accomplish some of these goals, we need a cross-disciplinary team. And this allows us also to use a variety of technologies, anything from looking at glia-neuron interaction and trying to understand how circuits are built and how the maturation is disrupted by aberrant synaptic pruning of circuits. We can use imaging across species. I'm showing you here human imaging from Mike Yassar's lab. And we can use big data and epigenomic approaches to really understand some brain and peripheral mechanism and identify biomarkers. We're using it, as I mentioned, through team science. But our approach to team science is not all these critters facing inward. Our approach to team science is inclusive. We have a center that is also a platform for a number of exciting and novel and important studies. And we invite you, everybody in the room and at UCI, to really be part of that. And how do we do that? Well, we believe that team science in the true sense of the word is also inviting other teams and incorporating other teams. And we do that in the Conte Center through monthly presentations where we not only discuss progress, but also hear from other people or discuss specific topics from different disciplines that change every month. We have seminars by real luminaries. The one that I'd like to draw to your attention to is that in the symposium on February 25th, the director of the National Institute of Mental Health, Dr. Josh Gordon, will be giving the keynote talk. So if you want to see, hear, and hobnob with Dr. Gordon, show up, you're invited. We also have seed grants, and I'm highlighting that because we just had our first call, and we got an amazing number of truly outstanding grants, and we'll be able to fund them at a better rate than NIH funding rates, so pay attention to the next round, which will be in a year or so. We really are interested in collaborations. We've been involved in generating preliminary data for proposals. We have tools, not only imaging, but also the entropy, the human behavior, quantitative protocols, questionnaires that are now going all over the country, and actually some of them around the world, such as the questionnaire about the unpredictability in childhood. And we have ears. We really want to hear from you. We really want you to be part of the team science that is the Conte Center. So with this in mind, again, this is what we think is team science in the Conte Center, and you are welcome. Thank you.
All right, and we have a slight change in schedule. So our next speaker will actually be Dr. Marcelo Wood.
Okay, great. So sorry about the change in the schedule. I was teaching earlier, and then I have to give this talk and then go teach in a different class right after this. So I wanted to start with a quote, and I think we've heard a lot of quotes, and I hope this one hasn't been used yet. But everybody knows about famous scientists, but here are a couple of famous space explorers, Captain James T. Kirk and Captain Jean-Luc Picard. And this is in reference to Mike Yassar, to boldly go where no one has gone before with regard to the Brain Initiative. So congratulations on all of this. Well done. So I'll say a little bit about the Center for Addiction Neuroscience. I'm the director. Francis Leslie, the former Dean of Graduate Division, is the co-director. And we've been slowly putting together the what we can from the addiction community to pursue one of the biggest problems facing the United States and around the world. So it's a major health issue. The cost is roughly $740 billion a year, which is absolutely staggering when you think of that number. There's been a dramatic increase in vaping of nicotine, and this is the biggest one-year increase ever seen for any substance in history. So again, mind-blowing. We're now in a time of legalization of marijuana, and yet we know almost next to nothing about what marijuana does to the adult brain, let alone the adolescent brain, and the consequences of development, as we just heard from Dr. Baram, from adolescent through the lifespan. And as we all know, there's a huge epidemic, the opioid crisis, and deaths related to opioids and the opioid derivatives, such as fentanyl. And all of these drugs of abuse impact the reward circuitry of the brain. The reward circuitry of the brain has evolved over millennia to drive goal-directed behaviors in humans. So it rewards us, so to speak, when we do things that are good for us, such as sleep when we need sleep, food when we need food, sex. Those things are all very rewarding, and the brain releases neurotransmitters to allow us to be rewarded for that behavior, so we do it again. And those behaviors are good for our individual survival and the reproductive success of the species. And so that's what this circuitry is important for, and drugs of abuse hijack that circuitry. And so things that might have been very rewarding, like food, sex, sleep, in the face of a drug of abuse, those drugs activate that system, and the effect on the neurotransmitters in the brain are massive compared to natural rewards. And so the brain is acutely aware of this other thing that is absolutely magnificent, right? And so the brain starts to want it more and more. It engages the prefrontal cortex in decision-making, so you go pursue that. And eventually, the brain starts to recognize that this is perhaps toxic. It starts to withdraw the release of neurotransmitters. And so natural rewards then start to become kind of pedestrian. They aren't drivers that they should be. And we forego sleep, we forego food, we forego things like that. And eventually, the brain starts to go into tolerance and withdraw. And then symptoms of withdrawal begin to appear with real severe craving. And then there's this kind of cycle with periods of abstinence and then periods of relapse where the brain is being driven to pursue these drugs, even in spite of very harmful consequences. And that's really how addiction is defined. And so at UCI, UCI has a tremendous history of neuroscience, and obviously there are very amazing scientists that are working in addiction within neuroscience. But in 2015, we brought that whole community together. We brought our colleagues from UCLA as well, and we basically discussed how do we do this better together instead of individually. And so in 2016, we formed the Center for Addiction Neuroscience. 2017, the Office of Research here at UCI provided some terrific pilot funding that allowed us to do a symposium, seminar series, pilot awards, and really allowed us to congeal. Things that developed from that were this NIDA P50. So this is a center of excellence. It's the flagship funding mechanism at NIDA, the National Institute of Drugs Abuse. And this one's called Impact of Cannabinoids Across the Lifespan, directed by Daniele Piomelli and involving quite a few faculty here. And now, 2019, we've submitted a training grant for the Neurobiology of Addiction. We have a course on addiction for our graduate students and postdocs. We're working on getting an even larger center status called ORU. And we really hope that in the near future we'll be a premier site of addiction research in the United States here at UCI. Leveraging the excellence in neuroscience that we have here. So ICAN, this is the kind of core group here, but there are faculty from 11 different departments, 6 different schools already involved, and this group has been putting on an annual research symposium. There's been a couple of public ones as well. There's an ongoing seminar series, the first addiction course, a journal club, small pilot awards that helped get us the P50 there, and we've submitted the T32, and just recently this week with Steve Baram, Christy Fowler, Dr. Tallie Baram, myself, and Vivek Swarp, we put in a U01, which is another funding mechanism looking at opioids. The P50, this is the Center of Excellence looking at THC. This is the psychoactive chemical in cannabis, and trying to understand how that affects the adolescent brain and subsequent effects on the adult brain. And so this is headed by Daniele Piomelli and heavily involves Steve Mahler, Chris Gall, Gary Lynch, a little bit of myself. And so there's a terrific animal and analytical research core, which is really critical to having rigor and reproducibility in the research that is being done, because this type of research on drugs of abuse can be highly variable, and we need to get rid of that variability so we can understand what's going on. There are public science events to increase the public scientific literacy, especially around these drugs of abuse that we know very little about. There's some key pilot awards that brought in 4 new faculty from 3 different departments in 2 different schools, and again, a seminar series being supported by this as well. And so these groups are all coming together, and our hope is that not only do we understand drugs of abuse, this compulsive nature of addiction, the reward circuitry, et cetera, but then we can also leverage that information for therapeutics and for once and for all make real headway in destroying substance use disorders. And so with that, thank you very much.
Up next, we have Dr. Tenner. Before Andrea comes up, I just want to mention this is a good juncture just to kind of, one of the themes that you're seeing across all of these projects is the open and collaborative environment. So none of the centers, none of these entities, and you'll see this again in Dr. Tenner's talk and any of the others, they're intended to be really open. So these are centers for faculty, by faculty, for students. So if you want to be involved, feel free to reach out, get to a seminar, get to a talk, get yourself on the mailing list. These are entirely open platforms, and I know that they'll echo the same sentiment as well. So with that, Andrea, please come on up.
Okay, so yes, thank you very much for letting me present a little bit about what Model AD is. And Model AD really means Model Organisms Development and Evaluation for Late Onset Alzheimer's Disease. So a big mouthful, but really very much like what Marcelo was talking about. We have Alzheimer's disease, which has been talked about as being the 21st century plague, and it really is a major disease because in the US alone it has 5.8 million people that are affected by it presently, and it costs $290 billion per year in care cost. So that means 1 in every 5 Medicare dollars is going to AD care. In addition, if we don't find some kind of way to stem this or slow the progression of the disease by 2050, there will be 14 million affected in the United States alone at $1.1 trillion, i.e. 1 out of every 3 Medicare dollars as it stands now, would go for Alzheimer's disease and Alzheimer's disease-related disorders, dementia care. So Alzheimer's disease is a progressive neurological disorder, and it presents first with a loss of cognitive ability. For those of you that don't know, they have the pathological hallmarks in this human brain are amyloid protein deposits, and what looks like here on these black arrowheads is dead neurons. Those are neurofibular tangles, or tau, hyperphosphorylated tau. You can see here that an AD brain, this is a human brain, has a lot more crevices and a lot broader versus the control brain, and that's because there is a loss of neurons, and it is the loss of neurons that causes that really is what's the cause of the dementia. But what's causing the loss of neurons? And so what we also see in the pathology is there are these cells that are in there that become reactive, indicative of inflammation, which are microglia and astrocytes. And so in order to investigate some of these diseases, these really complex human diseases, there's lots of different approaches one can take, particularly with human samples, looking at the clinic, imaging, tissues, and cell models. But what we're doing is looking at animal models, and that's the one we're choosing because here, by studying animal models of a human disease, you can test drugs in those models before you start testing them on humans, which would be a good idea, we think. So this is what Model AD is doing now. In order to think about getting a model, an animal model of a human disease, you have to sort of consider what you have to do. And it's likely that we're going to have to humanize some of these mouse genes, and particularly those that are key to AD, like an amyloid gene or the tau gene, to overcome some inherent differences between the human and the mouse. We could talk about that later if you are interested in that. In addition, there's multiple pathologies that you see in humans, and it may be that not all of those pathologies will be seen in one mouse model, so you're going to have more than one mouse model. Finally, there's also genetic effects, there's aging, there's the environment, all of these things have to be considered, and then there is a PC to MAC deficiency here. But nevertheless, a mouse strain background and the very complex genetics that all of us have. We are not inbred, which makes it very hard to work on us. In addition, very few of our human participants will donate their brain for while they're living for analysis. So what we can find out, though, is stuff about the genetics. And we know that a lot of the animal models that have been going on before were based on genetic mutations in these 3 genes. It doesn't really matter what they are, but they do cause Alzheimer's disease, very early onset, and pretty much 100%. They are rare mutations, but the risk of getting Alzheimer's disease is very, very high. However, that comprises only about 1% to 2% of all the individuals that have Alzheimer's disease. So what we do know that there are a lot of other genetic factors that have much lower risk conferred when you have these genetic variants, but they can be very common. Some of these are up to 40% of the population have those. Now, these do not cause Alzheimer's themselves, but in combination then could be factoring into the onset and progression of the disease. So what's Model AD doing? We want to prioritize these different variants for late onset Alzheimer's disease so that we can put them into an animal model and create these new genetic mice with the new CRISPR mechanisms that you might have read about. But we're using some old-fashioned recombination methods as well so that we put these genetic mutations into the mice. But what we have to do is select from this large number of possible genetic mutations, and we have to do that. We have to prioritize that. And so we look at how many studies do you see these variants coming up in Alzheimer's disease? Is there a predicted effect on the function in Alzheimer's disease? Is there a differential expression of those genes in Alzheimer's disease? And how close are the mouse and the human genes so that we can manipulate them without too much trauma to the mouse? And this is are there non-coding variant effects, which we won't need to go into. But once we have prioritized these, and you can see that these are in a number of different kinds of pathways, we can then begin to characterize those mouse models once we have put them into the mice and see which ones of those, which kinds of characteristics will come back and look like the humans. And what I mean to say, and this we've seen in the previous test, is we're going to take some of these mice, the new mouse models, we're going to look at RNA sequencing and the different omics. Does any of those look like any of the people that have Alzheimer's disease? If we look neuropathologically, do any of these mice that we have here, do any of those look like some of the people with Alzheimer's disease? And again, in brain imaging, can you do this? So these are a lot of different kinds of technologies, and so you need to have a lot of different expertise. Once we find a mouse, the red mouse that looks like the red person, the red Alzheimer's person, we can use different drugs in those particular mice, see if they are going to be protective against the disease, and those then would be candidates for use in those red Alzheimer's disease population. All of this is based on open science. All of the models that we generate are freely available to anybody without any legal restrictions. There's no patents, there's no licensing, there's no nothing. Everything is out there. All the data that we get is also open sourced, and you can get it through the websites. So what are our deliverables? We want to do 50 new mouse models of AD. We're about halfway there now and a little bit halfway through the grant. We've already got a mouse that has human A beta. We're working on the human tau now. And when we then put these other variants onto this platform mouse, we're going to test them in various diets and various kinds of environments to see if that makes a difference in speeding up Alzheimer's disease. The whole goal, again, is to share all of these mice models and the data that we get from them with other people to try their good ideas out so that we can get rid of this disease fast. Here at UCI, we've also then leveraged this whole thing by bringing in multiple other people into the group here. Ian Smith, for example, and Andy Obanas has come in, and a number of other people, Katrine Whiteson, doing different things on these mouse models, trying to characterize and trying to look at different factors. And we've actually then been able to bring in addition to the core grant, an additional $2.8 million in supplemental support in the 2 years that we've had this grant. So that's good, and that gets everybody then working together and makes it very exciting. So what does it take for this? This is our core team, so to speak. Frank LaFerla is co-director with me on this whole thing, but we really need a synergy of these different expertise across these different team members, and they all have different capabilities. Institutional infrastructure, like a computer. Yeah. And then also communication is very, very important. And what's really important in all of this then is mutual respect of the team members and their capabilities, diversity of opinions, and diversity of individuals coming in with different ideas, transparency, no hiding, everything is open, and as Marcelo once said, fearlessness. So you just get out there and do it, okay? So this is our team here. It's great fun. This is the rest of the team that do a lot of the work here. But we also work in conjunction with other labs, Indiana University, Jackson Laboratories, and SAGE, which are also in this Model AD consortium. We have actually bi-monthly meetings with them, weekly meetings with our NIH program officer, and we have an annual external advisory board of very distinguished individuals. All of these people then are trying to work out so that we can get rid of this Alzheimer's disease and be able to sustain our health care. So thank you, and if you have any questions, I'd be happy to answer them.
Thank you, Dr. Tenner. Are there questions for Dr. Tenner?
Yes.
Can you say a little more about how you go about focus work and sharing your data and models?
Okay, yeah. So the models that we make, the mouse models, we ship them to a facility called Jackson Laboratories. That's a commercial laboratory, and they breed them up and check them to make sure that they are the right background, and then they sell them. So they're not free. But there's no legal restrictions. You don't have to pay a license fee to UCI or to anybody. And commercial groups can have it, so academics can have them. That's for the mice. And for the data, I guess I didn't put the website up here, but if you go on to Model AD, it will give you directions to the AMP AD portal. And that's the Accelerated Medicines Programs for Alzheimer's Disease that is sponsored by NIA. And as soon as when we characterize our mice, all the RNA-seq data is uploaded, all of the immunohistochemical data is uploaded, the behavioral data is uploaded, LTP, that's all going to be uploaded and in that portal. Now, it might not be easy for a 2-year-old to do, but, you can get to it.
A little bit more than that. Okay, we have to move on. Thank you so much, Dr. Tenner. And up next, we have Dr. Zick Pizlo from Cognitive Science.
Oh, hello, everyone. I have to be in another place on campus in 15 minutes, so my talk will be even shorter than planned. So I'm in Cognitive Science Department, but today I represent primarily the Institute for Mathematical Behavioral Sciences. The institute existed for a number of years, but this coming academic year, the institute is trying to define a new theme for the institute. And the title, the name of the theme would be something like artificial mind. So what is artificial mind? You can think about artificial intelligence, but a little bit more specific. So this would be this aspect of artificial intelligence which tries to emulate the human mind. So one of the first steps would be to establish stronger relations between the cognitive sciences and School of Information and Computer Sciences on campus, because they are interested in artificial intelligence. But since here today we are talking about the brain, you could ask, why not building artificial brain rather than artificial mind? Or maybe it doesn't matter. And it seems that the two concepts, artificial mind and artificial brain, or mind and brain, they actually do refer to different things. They are like different views of the same stuff. And I don't have a lot of time to elaborate on this, but let's see how far we can go. So one of the, assuming that I can go to the next slide. All right, is to use the concept of what is referred to as strong AI. I'm not advocating this concept. I'm not saying that I agree or disagree with it, but looking at how the concept is defined allows you to see the difference between mind and brain. So strong AI claims that one can emulate all cognitive functions, all human intelligence on a computer, and it doesn't have, and it can, so you could have cognitive functions operating in a computer outside of the human brain. So this position assumes that there is nothing special about the biological nature of the human brain. It is an open question, what is the relation, and how far the work on mind informs the science of brain and vice versa. So what are the cognitive functions that we typically discuss in our institute and in my department? It would be visual and auditory perception, problem-solving thinking, linguistic communication, and so on. Obviously, you can immediately think about the brain circuits that are involved in those cognitive functions. So this is so at some point, it may not be obvious whether you need a clear distinction between mind and brain. One concept from applied mathematics which actually can help establishing bridges is the concept of optimization. Essentially, all modern theories of cognitive functions in psychology take the form of optimization problems. And that's nice because engineers are familiar with the concept of optimization, physicists are familiar with this concept, and biologists as well. So as one example, and I only have a couple of minutes to briefly describe this example of cognitive function, is visual perception. The world is three-dimensional, but the input to the human brain and to the computer is a two-dimensional image, camera image or retinal image. So mathematically speaking, there are always infinitely many three-dimensional interpretations that could have produced the two-dimensional image. How is your brain able to figure out which of those infinitely many is the correct one? And we now know how this is done. Your mind or your brain is asking which of those infinitely many three-dimensional configurations is maximally symmetrical. And then you actually, if you look at the cube, you see a cube. And this generalizes to many other objects because all natural objects in our lives are symmetrical, either because of the reason of locomotion or because of the reasons of growth. So this is how our algorithms actually work. We implement the algorithms, and the summary slide, we already have a good idea about how you could emulate human visual perception in a computer. So you could actually say that we were able to solve the strong AI version of visual perception. So what's the role of brain in this context? So one of the interesting aspects is that when you look at the algorithms which solve vision problem, it takes forever for them to analyze images. It is not unusual that the analysis of a single scene may take like 24 hours, but the human mind or human brain is able to analyze the three-dimensional scene in a fraction of a second. So it is tempting to think, as what the third bullet says, is that actually the biological structure of the brain, by the way of implementing the familiar least action principle in physics and chemistry, this may be a way of producing very quick solutions to optimization problems. So this is one way to see how cognitive science and how study of the human mind may actually interact with the study of the human brain. And that's the end of my talk. If you have questions, send me emails. Thank you.
Dr. Pizlo is very efficient. He's figured out the efficiency problem and solved that in AI. Okay, up next we have Dr. Chang.
Thank you, Mike. Congrats, of course, to this brilliant and amazing initiative and really putting together a great group of people here and a lot of very interesting talks. So I'm going to follow now with a little brief primer about what our group has been working on in the world of AI and precision health. Now, I will say I'm representing a team of 3 other individuals, Daniel Chow, Leslie Thompson, and Suzanne Sandmeyer. I oftentimes get thrown into these kind of things because I'm the single person that sort of straddles the different spheres of data science and computer science and medicine. So anyways, you guys get me today, but hopefully we'll, this is basically reflective of everyone's hard work. Now, I will start by paying tribute to this idea of team science, right? Like my colleagues before me, by just highlighting a little bit of sort of our vision and the way we've sort of built this new center. Now, very broadly speaking, the goal is simply to develop and integrate new forms of AI and machine learning into healthcare with a focus on that translational component where we put tools into the hospital. I'm a physician, right? So at the end of the day, what I want to see most out of my tools is for them to actually make it to real life and help a patient in some way. So that's very important. Now, the first question we oftentimes get is, what's the point of even building an institution or center to begin with, right? After all, most people that do this work, right, you'll have an umbrella of several PIs and everyone's sort of doing their own thing, building models. It's not immediately clear why or what role an institutional center may play. Well, what makes us a little different is that instead of that traditional model, instead of me kind of coming up with ideas and thinking what might be useful or interesting to make, all I house is a team of data scientists and engineers and hardware, right? And I ask really the rest of the community to come up with problems, right? I'm a physician, but really my colleagues in the other clinical specialties are the ones that know what's most valuable and most important. So I rely on them completely to come up with all the interesting questions. And so our goal really is just to lower the barriers for large-scale impactful AI work to help everyone else and enable the other teams out there to add a little bit of AI flavor into what they do. So what does that usually translate to? Well, again, instead of a single idea or 2 ideas that some of my colleagues might have who, again, it's not impossible to build AI algorithms, right? Over time, they will find a way to get where they're going. But hopefully, right, with sort of the infrastructure and the team and the people that we've set up, we're able to translate on those ideas much more efficiently, right? And really see these tools into practice. And really what these main pillars oftentimes involve is providing some degree of infrastructure support and or knowledge, right, to help people actually tackle these problems with AI and deep learning. Now, I will quickly point out a little note here on infrastructure, right? So it's not exactly clear sometimes what that might entail. And for those of you that might be setting out on your first deep learning AI project, you might think, well, okay, I'm gonna just be spending a lot of time writing code, right, building that model, running training and validation iterations. But the truth is, in medicine, if you want to do this properly, you're going to have to solve all these other things, right, that don't even involve building a model. Right, there are things like simply extracting data from our hospital system, right, anonymizing that data and putting it into a sandbox that you can do research with in a safe way. On the flip side, after you've developed the model, right, how are you actually going to take that model and use it in the hospital, right? What are all the different pipes that need to connect in the right way? What are all the regulatory hurdles that you need to think about, right, to make sure patients aren't being put into harm? So that part is extremely challenging, I will say, and certainly probably too much for a small team to really tackle on day one. However, what you'll notice is that all that infrastructure, as complex as it might be, is extremely reusable, right? Only the part where you build a new model is what you exactly have to change every time you have a new project. So that was our vision, right? Why don't we just build all of that once, do it well, and offer it to the entire community? Right, so again, if you take nothing else from this, I will say that this is an infrastructure that is available to the community. The imaging component is probably the strongest part of it, but we have other non-imaging data available as well. But the bottom line is if someone out there needs access to 10,000 patients with some sort of disease process or tumor or stroke, right, you guys can come to us. We'll get that data for you in just a few days. And if you need help building the models and doing downstream things, that's what we're there to do, to help out. All right, so what are some things that you can do with some of that infrastructure? So here's an example of a few tools. These two I like to highlight in terms of evaluating stroke, so looking for hemorrhage in the brain and looking for an inclusion in a proximal part of the circulation here. Now, what I like about this example is that you see in real time the diagnosis, right? The finding itself, while it might be pretty obvious, right, may not be the most complicated part of the process, but the fact that you had so many patients, right, waiting there in the ER, waiting for a diagnosis, the fact that you can have an AI system go through and automatically do that quickly and identify which patients need the most attention, right? That, in fact, is very valuable. Oop, it's going again. All right. Similarly, right, pathology has a very common theme as well, right? It's not impossible for a physician to go through and, estimate cell density or count nuclei, right? And a few high-powered field views, but it would be impossible to do that throughout the entire specimen. Right, it's too tedious, too labor-intensive. That's something really only a optimized machine learning model could tackle. And same with things in, for example, optical coherence OCT world. These pathologic specimens, right, there's probably a few thousand images that are being generated from the machine. So even though any single image could be evaluated with a human without any aid, trying to evaluate an entire stack of thousands of images could be very challenging. Now, of course, the power and the lure of AI is to try to do things humans cannot do, right? And so here's a few examples of that. In this project, we trained an AI system to look at raw CT data, which is a relatively low tissue contrast type of modality, and try to predict which areas have infarcted, right? Which areas the brain tissue has died based on some gold standard MRI imaging. And what the deep learning model was able to extract and essentially understand is how perfusion, right? Perfusion dynamics in different parts of the brain at each voxel correlate to a tissue infarct, and was able to actually generate these maps with a reasonable degree of fidelity. Here's an example, now moving into the world of brain tumors, where we train an AI system not only just how to identify mutations, like whether they could predict them, right? But also to show us what it learned after it finished training, right? After looking at hundreds and hundreds upon tumors, the deep learning system was actually able to go through and pick out why was it that it thought the patient had an IDH mutation or a 1p19q co-deletion, and generate really the first atlas completely curated by machine learning algorithm, highlighting the variety and heterogeneity of brain tumors. And then moving even further now into the world of personalized care and personalized medicine, this is an example of a project looking at treatment of brain tumors. So patients that are undergoing treatment, oftentimes the common problem we have is assessing whether or not something we see is recurrent tumor or just treatment effect. And instead of the traditional paradigm, which is to binary kind of split those two together, our vision here was to use a biopsy calibrated model to estimate the ratio, right? A continuous number of how much tissue is truly necrotic and how much tissue is viable tumor, really providing a very personalized metric on a patient-by-patient basis to make treatment decisions. And finally here, my last slide, those of you in the audience that have been paying attention, you might have seen now slides from radiology world, from pathology world, and the clinicians out there certainly are thinking about those, straightforward demographic risk factors and non-imaging covariates. But of course, right, the real sort of goal of all this one day is to combine all that data. Right now, we do a lot of analysis on a single modality because it's very challenging to go across the different worlds. But in fact, with deep learning, with sort of the latest machine learning models, this is something that's very feasible. So anyways, that's all I've got here. This is my contact information, and more importantly, the information about our center. Again, the invitation remains. Anyone out there that wants to kind of chat and work a little bit more, feel free to reach out. Thank you.
All right, and our final speaker of this session is Chair of Psychiatry and Human Behavior, Dr. Ruth Benka.
Okay, well, I think I'm last for a couple of reasons. I could say the best is last, but we're probably last because we're the youngest center. We're sort of still a work in progress, and we're going to tell you a little bit about the progress today, but also probably because by this point of the afternoon, you're all asleep. So we'll try not to disturb your napping too much. Okay, so the Center for Sleep and Circadian Neuroscience, as I said, is a work in progress. And I think one of the important features that we have is we actually have a new building. So the new Clinical Sleep Center, which was really built to be a clinical and translational research center, is at Birch Street, about 5 minutes from here. And we have the whole first floor of this building, and it's a beautiful sleep lab, and I'm going to show you some of the features of it. But let's talk about why sleep and rhythms are important for UCI Brain. And that's because sleep is really integral to every life form. And if you think about circadian rhythms, pretty much everything has a circadian rhythm, even single-celled organisms like algae. In terms of the human situation, and actually most animals that have brains, we know that sleep is required for brain plasticity. And sleep and rhythms together organize the regulation of our synapses. We used to think, and to quote Alan Hobson, that sleep is of the brain, by the brain, and for the brain, but we now know that sleep is really important for all aspects of physiology, both within and outside the CNS, and it's critically important for health. To just talk about the relatively new history of the SCN Sleep Center, or the SCN Center. UCI has been known for its strengths in circadian neuroscience. We have several really eminent scientists here, Paolo Sassoni Corsi and Todd Holmes. We have other sleep scientists here on campus, and one of our missions here is to try to bring everybody together and actually have everybody do sleep research. And one of the reasons I came here a few years ago was that this was one of the only academic centers that I was aware of, actually the only one that didn't have a sleep medicine program, so it was a great opportunity. And two important things happened, one of which is in 2017, we got a seed grant from UCI to help develop this center. And secondly, we built the new center in Newport Beach, and we built it so that it would have state-of-the-art capabilities both for research and clinical work and be an opportune site for people to do translational research. So one of the things that we have is we have high resolution or high density EEG. We can do 256 channels of EEG all night long on people or during the daytime, which is really important because we need to start thinking about sleep as not a one-dimensional behavior of like how many minutes did you sleep or how much time did you spend in a particular sleep stage, but to understand that sleep has 3 dimensions in the brain. Not all parts of the brain sleep the same way at the same time. And furthermore, sleep has a temporal component. Your brain is different when you go to sleep at night and when you wake up in the morning, and stuff is happening all night long, and we have to be able to look at it. So we like to use high-density EEG across sleep as a brain imaging tool. It's probably it is the brain imaging tool that has the best temporal resolution. We can sample at 10,000 hertz if we need to. Here we have in the bottom right a picture of one of our eminent sleep scientists, Dr. Bryce Mander, in an electrode cap with one of our technicians. And when we put on the electrode caps, we can look at important sleep waveforms on the upper. Do I have a pointer here that works? Maybe not. Yeah, there it is. Okay, so that's where sleep spindles are most prominent in the vertex, and here are slow waves in prefrontal areas, and spindles and slow waves are thought to be critically important for some of the functional aspects of sleep, and they only occur during sleep. Some examples of other things we're trying to do in the lab is we're partnering with the Beckman Center and Hamamatsu to add oximetry, basically brain oximetry using near-infrared spectroscopy. This is Rob Warren, who is the postdoc who has been working on this. And because typically what we do is we look at pulse oximetry. And this is what we do in sleep labs and most sleep research. However, what's going on in your finger may not really be very representative of what's going on in your brain. It's really your brain that needs the oxygen, not so much your finger. And in fact, the patterns of oxygenation look very different in the brain. This is basically a number of minutes. We're looking at about a 15-minute period here. This is an apnea where someone has stopped breathing, and we can see following the apnea, because it takes a while for the blood to get from the lung to the finger, we see a drop in blood oxygen level. But here looking at with near-infrared spectroscopy at the prefrontal cortex, we see a very immediate and dramatic drop in oxygen level. My interests have been largely on looking at, more recently, at local changes in sleep topography related to a variety of neuropsychiatric and sleep disorders. And here's one example of one of the most common sleep disorders, sleep apnea. And what we see in comparing groups of individuals with sleep apnea to normal controls is that there is a decrement in slow wave activity in these posterior regions of the brain, which actually map onto the posterior cingulate cortex, and this is in the absence of an apnea. And the severity of this loss of local slow wave activity is related to the severity of apnea. Now, the reason this is quite interesting, we've been talking a bit about Alzheimer's today, is that sleep apnea is a major risk factor for Alzheimer's disease in terms of earlier onset and more rapid progression. And if we actually look at slow wave activity in groups of individuals who are at risk for having Alzheimer's disease because of family history or ApoE4 status, but do not have sleep apnea, we generally see that they have these decrements in slow wave activity in similar areas of the brain, as well as some decrements in frontal areas. This raises the issue is that if apnea is also causing decrements in these areas, and we know that sleep is important for clearance of amyloid and for decreasing production of amyloid, this may be part of the link between apnea and Alzheimer's disease. And right now we're doing studies to look to see if treating apnea slows Alzheimer's progression in people who are asymptomatic but amyloid positive. Bryce Mander, I'm going to now run through a couple of our faculty that have started doing work in the center or related to the center. So this is our high-density EEG team. This is Bryce without a cap on, and some of his staff. And they've been finding that, again, in individuals at risk for Alzheimer's, that losses of spindle activity, and we think spindles are also important for learning and memory, loss of spindle activity is correlated with tau deposition. We are, let's see, did I skip something? Okay, no. All right, next, this is one of our new graduate students, Miranda Chow-Farley, who is very interested in sleep and exercise, but has been starting out by looking at, in collaboration with UCI Mind and the Consent to Contact registry that they have of several thousand individuals. She's found that people who have subjective sleep problems also have memory complaints, and that the people that sleep either less than 6 hours or more than 8 hours have more memory complaints than people who are sort of in that golden spot of getting at least 6 to 8 hours of sleep at night. Another investigator, we're working with some of the scientists in neurobiology and behavior, and to follow up on Andrea's talk with some of the mouse models, we're trying to see how mimicking sleep apnea in a mouse, we can't make it snore or stop breathing that way, but we can expose it to intermittent hypoxia. Well, actually, Kim can, and we're trying to see whether this intermittent hypoxia is accelerating Alzheimer's pathology, and this is something we can do with these various mouse models to try to understand some of these links. Similarly, Norbert Fortin is looking at sleep fragmentation and trying to understand some of the neural mechanisms of why intermittent hypoxia or sleep interruption may be contributing to Alzheimer's pathology, again, in rodent models. Mark Mapstone is working on looking at metabolomics and inflammatory markers that may be increased or altered with sleep disorders or sleep disruption or sleep deprivation, and how those are contributing to neuronal dysfunction and neurocognitive effects. So a lot of work that we're doing related to normal and pathological aging, but we're also very interested in children because sleep is, of course, extremely important for neurodevelopment. And in collaboration with PERC, we are looking at the relationship between sleep and aerobic fitness. So we know that teenagers, of course, need to sleep, they need to exercise, they need to eat properly, et cetera. And so one of the questions is, if you exercise but don't sleep, do you get the same benefits in terms of cardiovascular risk factors and also improvement in your fitness as if you exercise and sleep well? Conversely, if you sleep but you don't exercise, probably that's not going to be as good for you either. And so we have an opportunity to start to look at these interactions. And going even earlier, we're working with the Conte Center and Tallie's group, who has collected just some amazing data on sleep during pregnancy, postpartum, and in children as they grow up. And so some of the questions we're interested in is, how does mom's sleep affect their children's sleep? And how does the sleep of children affect their mothers and vice versa? And that gets into this idea of disruption and disorganization of behavior, because sleep is, of course, a behavior that needs to be well organized. And so far, what Ivy's found is that controlling for the level of depression, mothers who are not sleeping well while they're pregnant tend not to sleep well after they give birth, and they have higher risks of postpartum depression. And this is another example of where disturbed sleep actually contributes to psychiatric disorders. So this is just sort of a sampling of what we're doing, and we're just really getting started. So we want everybody to think about sleep when you do anything related to the brain, because your brain doesn't function well without sleep, and we're all happy to try to help out. And we have a beautiful new lab, and if you have gadgets you want to test, or you want to come and do studies and use this equipment, it's really some of the nicest clinical research space we have at UCI now. And I'm going to finish up. This is a sleeping anteater, and to see if you have any questions.